First, we need to discuss who is responsible for wildlife health and what do we mean when we talk about wildlife health. For this presentation, I will assume most people watching will be biologists and will have some familiarity with the North America model where wildlife resources are held in trust for the public good. This means that the stewardship of wildlife resources falls to the natural resource agencies that have particular land or species under their jurisdiction. By holding these resources in public trust, these governmental agencies have to think about the current and future uses of wildlife by the public. That means that sustainability of wildlife resources for future generations is part of their mission. Therefore, we have seen that some wildlife diseases are capable of having population-level impacts and that agencies must respond as part of their public trust mission. So before we get too far ahead of ourselves, we should probably make sure that we have a clear definition of wildlife health. In this case, our definition feeds back into the sustainability and the role of wildlife in the ecosystem as maintaining the population levels that meet their functional roles for society and the natural world. Now, that population level can vary quite a bit if we're talking about a game species versus an endangered species. So why should biologists care about diseases other than their public trust responsibilities? We know that disease is a part of the natural ecosystem in some cases, but many of the effects those diseases have are more critical now because of the human landscape modification and globalization that have introduced novel pathogens. Disease is often most influential if we're dealing with populations at the edges of their habitat availability, such that density can become very high and facilitate disease transmission, or conversely for threatened and endangered species, where we're concerned that the population will get so low that it's no longer viable. As a biologist, when we think about the three demographic factors that influence populations, recruitment, immigration and emigration, and mortality, these are all things that we think about with disease issues, so it's a partnership that we must forge between wildlife health specialists, and I use that term generally, and field biologists. Other things to think about is that eradication of disease in free-ranging wildlife has not happened. If anything, there has been a resurgence of infectious diseases since the 1990s. Fortunately and unfortunately, biologists are in the best position to be informed, but also to be involved in transmission. When we talk about diseases going between animals and humans, the term we use is zoonotic. Just to drive the point home, this is a picture of Eric York. He was a biologist researching mountain lions in Grand Canyon National Park. He died of pneumonic plague in November 2007. He was 37 years old. One of his collared cats died, and he carried it out of the backcountry over his shoulders and did a necropsy in his garage. A few days later, he didn't feel well and went to the park's health clinic. They diagnosed him with the flu and sent him home. 49 people had been in contact with him before he died at his home several days later. While this story is tragic, I feel its message is critical to remind people that these things do happen, and we need to protect ourselves and be vigilant to possible disease issues in the course of our daily activities. Zoonotic diseases have brought forth new awareness to One Health, which recognizes that pathogens move between all animals, whether wild or domestic, and humans. It's great at bringing together human medicine and veterinary medicine with wildlife. Unfortunately, because the field of wildlife health is relatively young compared to others, there's a void where it often results in finger-pointing towards wildlife as the source of pestilence. Hence, wildlife are often portrayed as the villain rather than the victim in these scenarios. So why does wildlife draw the short end of the straw when it comes to One Health? I would say the short answer is because that it's not easy. First off, it's difficult to identify or predict circumstances in which disease may pose a threat because we don't have the same level of scientific inquiry and data collection for wildlife. We don't have the same ability to manage disease, often because there's jurisdictional issues on who is responsible for a free-ranging, unowned wild animal. It can depend on the species, the location, and the pathogen. Our abilities to respond to diseases using different tools and techniques have not been well developed, and often this has resulted from limited funding. Finally, because disease is typically overlooked until it's a major issue, there's little political or funding available to make necessary changes. We also deal with the public's desires and traditions that limit their willingness to make changes. Let's talk a little bit more about zoonoses. Remember, these are the diseases that are transmissible between animals and humans. About 60% of the known infectious diseases are zoonotic. A paper by Taylor et al. In 2001 reported that 175 emerging infectious diseases, out of those, 75 of them were zoonoses, and zoonotic pathogens were twice as likely to be associated with emerging diseases than non-zoonotic pathogens. The mode of transmission of zoonotic species is also more likely to occur through vectors, such as mosquitoes or ticks, or by indirect contact rather than direct contact. Some zoonotic diseases you may encounter during the course of your work as a biologist includes rabies, brucellosis, tuberculosis, avian influenza, tularemia, West Nile virus, Lyme disease, and eastern equine encephalitis. So let's talk about wildlife surveillance. It is the only way to know which pathogens exist in populations, in which geographic areas, and in which populations. The World Organization for Animal Health defines surveillance as the systematic, ongoing collection, collation, and analysis of information related to animal health and the timely dissemination of information to those who need to know so action can be taken. It is the single most important component of a wildlife health program to detect new and emerging pathogens. Let's review the key points of surveillance in more detail. First, it must be a continuous activity that requires vigilance and investigation. Rarely are you able to say that a disease is never going to be a threat, even for smallpox, which has been eradicated. In the United States, the threat of foot and mouth disease is so great that they regularly hold workshops to plan for the response. You need regular analysis of data for specific purposes and to detect trends because surveillance is expensive to conduct. For example, millions were spent on highly pathogenic avian influenza using three strategies: investigating mortality events, using hunter-harvested samples, and conducting live bird sampling. Analysis showed that more viruses were being detected from mortality samples than other methods, so the Interagency Avian Influenza Working Group recommended focusing primarily on mortality investigations. Your job is not done when the animal is tested. It's critical to communicate the results of that data collection, and this requires some synthesis and interpretation to understand that data. Finally, the most important feature is to distribute the analysis to the people and agencies that need it. Whether it's publicly available on a website or in a publication, we want to make sure that those reams and reams of information stored in file cabinets actually are available and useful. Without analysis and publication, it's not valuable. In any disease surveillance, there are two major forms. The first is passive, which may also be called general or scanning surveillance. This allows you to know which pathogens exist in your area of interest and be vigilant for emerging diseases. This is the kind of surveillance we do regularly for diseases like rabies and canine distemper. The second is active or targeted surveillance. With this method, you identify specific diseases or species. You have a defined sample size or cutoff, which is determined by probability-based methods to be able to declare yourself free of disease or determined time of first detection. You can plan how animals will be collected and tested ahead of time, and the strength of these methods are that you now have a valid estimate of prevalence and geographic distribution. This is the type of sampling that we use for chronic wasting disease in deer. The only problem is that since you are only looking for a particular disease, you typically don't detect new ones. So let's break down the components of surveillance. Obviously, for passive surveillance, you need detection of sick or dead wild animals. This is your most important source of information. Now, who finds them? It could be the general public, hunters, fishermen, birders, farmers, veterinarians. If these animals are collected, who collects them? Is it biologists, the public health agency, the ag department, wildlife rehabilitators, or veterinarians? Ideally, you would like people who spend time with animals and know how to collect them properly to keep themselves safe. If it's a large-scale mortality event, you may want a field investigation by wildlife disease specialists. The next question is, who are they reported or sent to? In New York, we have a cooperative wildlife health program between the New York Department of Environmental Conservation and Cornell University. We assist people by providing information on how and where to send carcasses and how to euthanize animals if they're sick and moribund. We make sure a network of field biologists are maintained in the different regions around the state to respond to these types of events. It's important to get samples to the lab in a timely fashion. So there's 2 different locations for large carcasses that need to be driven and not shipped overnight. The second component is identification of pathogens that cause morbidity and mortality. Because we are never quite sure what pathogens wildlife species might have, we prefer to work in a fully equipped diagnostic lab where the necropsy, or animal autopsy, is conducted under a biosecurity level 2 or 3. That means those potentially zoonotic pathogens are contained. The diagnostic lab at Cornell is staffed with pathologists, microbiologists, virologists, molecular specialists, and toxicologists. But even all of these people cannot make a diagnosis without having some history of disease in this particular species or area or time of year. It gives them clues on which tests to prioritize, and this is called the list of differentials. If you just have a dead animal, the list of differentials can often be long, and the presentation in wild animals can be quite different than what's typical in domestic animals. We also have very few tests developed specifically for wildlife, and because of this, we have to be cognizant that some of the tests may provide false results or have low sensitivity and specificity. Finally, the quality of the carcass plays a major role in whether or not we're able to conduct any testing. Because wildlife often have an unknown time of death and are exposed to challenging environmental conditions, plus you have the transport time to get the samples to the lab, carcasses don't get fresher if you can't get a specimen to the lab quickly. And by quickly, I mean in less than 48 hours. It's often better to freeze an animal to stop the decomposition. Autolysis is a fancy word the pathologists use to say rotten. The third component of surveillance is information management. Ideally, all information is stored in a long-term database to archive and preserve information over time. You want this database to be easy to search and map. This information should be disseminated to relevant parties, such as the person who found the specimens, the biologist, the public health agency, and others that are interested. Standardized information makes creation of publications, reports, and websites much easier. Finally, sharing the data across platforms, having interoperability is really the ultimate goal, so you have much better access to information about potentially emerging pathogens. Bringing the circle to a close, I want to emphasize interdisciplinary cooperation. This really is the underpinning of the One Health concept. You want to make sure that your partners have the necessary information so that appropriate steps are taken quickly. These partners are also beneficial because you can leverage the resources of one agency with that of another. Oftentimes, the factor that is limited here is mutual understanding or sharing the same language. Because biologists may not be well versed in the field of human or veterinary medicine, they're not always on the same lingo. But biologists have quite a bit of insight regarding populations of animals they study and should be at the table when discussing actions that could mitigate the disease. So why do we do wildlife surveillance? Well, the first 2 reasons are from the World Organization for Animal Health. First, we want to reduce the costs, be these costs to social, human health, economic, or ecological, to society from pathogens in wild animals. Second, we have an obligation to detect and report pathogens in wild animals, particularly because we are seeing an increasing number of emerging zoonotic diseases. Finally, this is the one I added, we want to ensure healthy wild populations through early detection and management. Healthy does not always mean at the density desired by the public, whether that's high deer density for hunting or low density coyotes in suburban areas, but goes back to the definition I used at the beginning of this presentation, where an animal is fulfilling its ecological role. So here is the schematic of what happens in a wildlife disease outbreak. Although an external observer may not see this way, wildlife disease surveillance is an organized system of observation in wild animals that incorporates knowledge from the field with detection of sick and dead animals, followed by sample collection, which those specimens are then sent to a veterinary diagnostic lab to identify pathogens, which may require additional field and laboratory research. We use information management systems for record keeping, data analysis, mapping, and risk analysis. And finally, communication, which may involve further training for field staff and outreach to the public. In conclusion, wildlife professionals need to recognize that part of our stewardship role is to conserve animal populations, which includes thinking about and reacting to wildlife disease issues. Disease emergence has been increasing in the past 3 decades and is predicted to continue along that trend. Disease can have an impact on wildlife populations. I encourage anyone in the wildlife profession to put on their futuring cap and think about how the conditions are changing for wildlife. What are the activities currently allowed that may have an impact on the future? Human behaviors are becoming more impactful on wildlife species, and we need to think about how to best protect wildlife as our public trust resource for now and future generations. Thank you.